Hi my dear students, welcome to my biology class. In the previous sessions, we have learned about the structure of a polynucleotide chain and the salient features of DNA. I hope you have understood the topics well. Today, let's familiarize with another topic, central dogma or basic formulation of molecular biology. We know that DNA is the genetic material in most of the living organisms except a few living organisms. It is a master copy of genetic information. When we have learned about the structure of DNA, I have told you that DNA is chemically and structurally stable because of the arrangement of nitrogen bases, the sugar and phosphate bond present in them, presence of thymine in them. The DNA is highly stable than RNA. This particular concept was formulated after the discovery of the DNA double helix by James Watson and Francis Crick. Later on, Francis Crick in 1958 proposed this particular concept in molecular biology. It states that there is a unidirectional flow of information from the master copy, that is DNA, to the working copy, which is RNA, and finally to the building plan polypeptide or protein. So, the central dogma in molecular biology can be diagrammatically represented as DNA is the master copy of information and it is able to duplicate or replicate, it is able to produce its copy. So, replication. And this DNA helps in the production of a corresponding mRNA by a process called transcription. Okay, this mRNA is the messenger RNA. Okay, this messenger RNA is able to express the information that is encoded in the DNA in the form of nitrogenous bases. The same information can be expressed in the form of 4 letters, 4 nitrogenous bases. Then from here, the information that is present in the mRNA finally will be translated into a polypeptide or a protein. So, by translation, it helps in the development or the production of a polypeptide or protein. So, what is the necessity of producing protein in living organism? A protein controls a particular trait in the living organisms. The information for synthesizing the specific protein is ultimately stored in the master copy, that is in the DNA. So, it is converted into mRNA by transcription. Then from the mRNA, it is translated into a polypeptide or a specific protein. This protein controls a particular trait. Okay, this concept is called central dogma or formulation, basic formulation of molecular biology. In the DNA, the information is stored in the form of 4-letter languages. 4-letter languages here means the nitrogen bases. They are adenine, thymine, guanine and cytosine. This 4-letter language is transcribed into messenger RNA, that is in the form of another 4 letters. 4 letters like adenine, uracil, guanine and cytosine. Here there is one difference. Instead of thymine, in the mRNA there is uracil. And these 4-letter languages, that means 4 different types of nucleotide chains in the mRNA is translated into a chain of amino acids. Okay. Chain of amino acid forms a polypeptide. Later on in 1970, 2 scientists named H.M. Temin and D. Baltimore discovered the reverse transcriptase in RNA viruses. Reverse transcriptase enzyme in RNA virus, a group of RNA viruses, retroviruses. They discovered that the reverse transcriptors perform the central dogma of molecular biology, reverse by synthesizing DNA from RNA. So here the genetic material of the virus is RNA, so that this RNA synthesizes the DNA by a process called reverse transcription. Reverse transcription by the enzyme called reverse transcriptase. Okay, this DNA again produces the mRNA by transcription and finally produces a protein. This happens in reverse transcription. This type of reverse transcription can be seen in the HIV, the virus that causes AIDS in human beings. Examples: HIV, human immunodeficiency virus, TMV virus, tobacco mosaic virus. Okay, one more example: QB bacteriophage. QB bacteriophage. So both transcription and reverse transcription takes place in the living organism. Most probably the transcription is the main phenomenon that takes place in most of the living organisms, as most of the living organisms have the DNA as their genetic material. I hope you have understood the topic well. With this, I conclude my session. Thank you and have nice moments.